Hi everyone, and welcome back to the Basics of Neuroscience course. Last time we covered the cells that make up the brain. Now in this lecture, we're going to be covering how these cells communicate with each other in order to send messages. Neurotransmission is the process of transmitting messages in the nervous system. So it's transmitted through what we call synapses. This is all made possible by the unique properties of neurons that we talked about last time. So they have the ability to generate electrical impulses that can then propagate across long distances from one cell to the next. So this connection is the synapse, which we're going to be seeing in the next couple of slides. So if we look at this diagram on the right, we can see that the axon is contacting the dendrite of the next neuron. So we said the axon is the output and the dendrite is the input. That's because the synapse from one axon of what we call a presynaptic neuron is going to contact the dendrite of the postsynaptic neuron at the synapse. Now the electrical impulses will take the form of what we call an action potential. So the action potential will pass through the first neuron, and then neurotransmitters, which are molecules that are essential for neurotransmission, will be released at the synapse to activate a certain response in the postsynaptic neuron. So this is just a brief overview, and we'll be going into the details of every part of the summary I just provided. Now, to understand how the action potential happens, we first have to understand the resting state of a neuron. So the word potential just refers to the difference in charge across the membrane in the cell. So a negative value means that the inside is negative compared to the outside. In a neuron, there are certain things that happen at rest that determine this relative charge. First, there are many intracellular proteins that are negatively charged. Second, there is a sodium-potassium pump that maintains the proper equilibrium of sodium and potassium ions inside and outside the cell. So what it does is it pumps 3 sodium ions out of the cell and brings 2 potassium ions into the cell. These are both positively charged, but if we compare the outside to the inside, the net charge is +1 outside due to the extra sodium ion. Now, it's important to note that in addition to this electrical difference, there's also a chemical difference that builds up. Since there are a lot of sodium ions outside the cell, some ions will flow into the cell down their concentration gradient, while the potassium ions will leak out of the cell since there is a higher concentration of the potassium ions inside the cell. So accordingly, if we measure the voltage inside the cell relative to the outside of the cell, the value is negative. So the resting potential of a neuron is actually around -70 millivolts. So what happens during an action potential is that we actually have a huge reversal in this charge due to an electrical impulse that will change all of the processes that we just mentioned that take place at rest. So the potential will go all the way from -70 to around +30. So there's a reversal in the charge, and there will be a positive potential during the action potential. So the process starts with a certain stimulus, for example, from another neuron, that will lead to the opening of some ion channels and the flow of positive ions into the cell due to the electric attraction with the negatively charged interior. So if this flow is strong enough to get the potential to -55 millivolts, which we call the threshold potential, then voltage-gated sodium channels will open, and then there is a huge influx of sodium ions into the cell through these channels, and this is the phase that we call depolarization. So the potential, instead of being negative, is starting to go towards 0, and then eventually it will be positive. So the potential reaches a peak of around +30 millivolts, and then the sodium channels will close and voltage-gated potassium channels will now open so that potassium can flow out. This stage is called repolarization, since the neuron is going back to its negative potential. Now, because the potassium channels stay open for a while, the potential becomes slightly more negative than it was at rest due to this major flow of potassium ions out of the cell. So this is what we see in 5 in the diagram on the right. So we can see it's a little more negative than -70. And this is actually the refractory period during which the neuron can't be stimulated, and so it can't have another action potential. This is because the potential is very negative. Now the sodium-potassium pump will work at this stage to re-establish the equilibrium by again pumping 3 sodium ions out and 2 potassium ions in order to ensure that the neuron will reach the resting potential again. Now sometimes the process of the action potential can seem really abstract, but what I would recommend is to watch a video on YouTube about the action potential, because just seeing it visually can really help in understanding the concept. So it's important to understand also that not every stimulation is going to lead to an action potential. So I said that if the neuron potential reaches the threshold of around -55, then we can have an action potential. But not all stimulation is going to reach the threshold. So weak stimulation at the synapse will just lead to small transient and small changes in the voltage across the membrane, so we call these graded potentials. In addition, there's a phenomenon called summation that can help the neuron to reach a threshold and trigger an action potential. So here in this diagram, we can see how this works. If we have multiple excitatory inputs, so these are inputs that are trying to push the neuron towards an action potential, then we're going to have the opening of some ion channels, so that the effect will be summative. So the voltage will go all the way from -70 to just below threshold. Now, if the neuron receives another small stimulation, then it will reach the threshold of -55, and then we have an action potential. So the action potential is considered to be actually an all or none event, which means either the stimulation is sufficient to lead to an action potential or it doesn't. There is no weak action potential or strong action potential. So the amplitude of the action potential is always the exact same. So another interesting thing to note here is we can also have inhibitory potentials, which are shown in the dark pink color here. So these are from inhibitory neurons, which we saw in the last lecture. So now you can understand what these neurons do. So instead of depolarizing the postsynaptic neuron, these inhibitory neurons want to push the neuron further away from an action potential. So these inhibitory neurons want to silence the postsynaptic neuron by pushing it away from the threshold and making it more negative. So they prevent action potentials. So now we know that at the synapse, some ion channels in the postsynaptic neuron are going to open in order to try to trigger an action potential if the stimulation is excitatory. So how does the action potential then propagate all the way to the axon terminal in order to then pass the impulse to the next neuron at the next synapse? So what happens is that the current flows through the axon, but in order to prevent leaking of ions out of the axon, since we said leaking of ions can happen, the myelin sheath surrounds it. So this is where the myelin, the function of myelin comes in. So it will insulate the axon in order to keep the ions inside, to keep this current. And also the myelin speeds up the neurotransmission because the voltage-gated sodium channels are only found in the naked parts of the axon, so those segments that we call the nodes of Ranvier. So the result is what we call a jumping of the action potential from node to node, and this is referred to as saltatory conduction. So instead of having to trigger an action potential at every single segment of the axon, the current just flows through the axon, and then at each node of Ranvier, additional sodium ion channels are open, and then they further propagate this impulse. So now, if we consider that the signal or the action potential or the impulse has reached all the way to the axon terminal, to trigger any type of change in the postsynaptic neuron, neurotransmitters have to be released. So these neurotransmitters are stored in small vesicles that remain in the axon terminal, ready to be released whenever a signal arrives. So once the action potential arrives, the change in voltage opens voltage-gated calcium channels. Calcium ions will then flow into the axon terminal, and they cause the vesicles to fuse with the membrane to release the neurotransmitters into the synaptic cleft by exocytosis. The neurotransmitters then can bind to their receptors on the postsynaptic membrane to cause the opening of ion channels on the postsynaptic neuron, and this will then lead to a change in the membrane potential of the postsynaptic neuron, and if it reaches the threshold, there will be an action potential, and the same process will continue to occur from neuron to the next. So to prevent excessive stimulation, the last part in this process is once the signal is terminated, the neurotransmitter is either broken down by enzymes in the synapse, or it is recycled by being reabsorbed by the neuron. So now that the synapse is at rest and all of the neurotransmitter is cleared, the synapse is basically ready for another impulse. So neurotransmitters typically fall under one of these three categories. They can be either single amino acids, amines, or neuropeptides. So the amino acids are the smallest ones and the neuropeptides are the largest. So here these are some of the most well understood and traditional neurotransmitters in the nervous system. So glutamate is the main excitatory neurotransmitter, and GABA is an amino acid that is the main inhibitory neurotransmitter. Serotonin is a famous neurotransmitter that is released in certain areas of the brain, and it is known to regulate mood, and we will go into it in more detail in the next slide. Dopamine is another famous neurotransmitter. It's also released in specific pathways and regions in the brain, and it is involved in reward, pleasure, and mood. Noradrenaline, or what we call adrenaline, is the main neurotransmitter that is used in the sympathetic nervous system in order to activate the stress response, or what we usually call fight or flight. Acetylcholine is mostly used in the peripheral nervous system at the neuromuscular junction, which is basically the synapse between a neuron and a muscle. And then basically each neurotransmitter will have a specific receptor that will activate certain signaling cascades inside the cell, but we won't be going into this in details. So here's the example of serotonin. And basically the pathways and the brain regions where it is released. So as we can see, it functions in many areas across the brain, and it is one of the neurotransmitters we hear about a lot because many medications such as antidepressants work via the serotonin pathway. So these drugs are called selective serotonin reuptake inhibitors, or SSRIs for short. And we can see their mechanism of action in panel B that shows us the synapse. So SSRIs will actually block the receptors on the presynaptic membrane to prevent the reuptake that I just mentioned. So therefore, this will allow serotonin to remain in the synaptic cleft for a longer period of time, which will basically increase the activity and activation of the postsynaptic neuron. So this is important for mental disorders in which serotonin is thought to be at lower levels than normal, like in depression. And these are some in panel C. You can see a few of the most common SSRIs. So when we covered astrocytes in the last lecture, I mentioned that glia were initially thought to play only minor roles of support to the neurons, but the astrocytes actually have been found to be really important in synaptic neurotransmission. This is a crucial function of astrocytes, and it led scientists to actually coin the term tripartite synapse. So this term refers to the astrocyte, the presynaptic neuron, and the postsynaptic neuron, as we can see in the diagram. So astrocytes are in such close contact with synapses because they regulate neurotransmission through multiple ways. So first, they can reuptake neurotransmitters, and that is important to avoid accumulation of the neurotransmitter in the synaptic cleft, and also to avoid spillover to adjacent neurons that are not intended targets of the neurotransmitter. This also prevents the overstimulation of the synapse, which can be toxic for the neurons. Next, the astrocytes are also capable of breaking down the neurotransmitters into subunits, and then it shuttles them, the astrocytes shuttle those subunits back to the presynaptic neuron so that the presynaptic neuron can then resynthesize neurotransmitters and package them into vesicles. So it contributes to the recycling of the neurotransmitters. And then finally, astrocytes actually, by monitoring the synapse, they're able to also regulate the ion balance, and this will ensure that action potentials can take place. In this final slide, I just wanted to show you some different ways in which neurons can link up to each other in order to give you an idea of how complex these connections may be. So first, this is an example on the left of competing inputs, and it happens when there is a neuron that is contacted by an excitatory neuron and an inhibitory neuron. So one will push it towards threshold to trigger an action potential, while the other, the inhibitory neuron, will push it away from threshold towards a more negative potential. So in this case, the presynaptic neuron that provides the stronger input will determine what the postsynaptic effect will be. So we saw this before when I talked about summation, so that's what will happen here. Now in the second scenario, in the diagram on the right, we have what is called disinhibition. The first neuron inhibits an interneuron that is also inhibitory. So since the second neuron is no longer able to inhibit the final neuron, then the final neuron will be able to fire. So if we take it in terms of summation and simple math, two negatives make a positive. So that's what represents disinhibition. When we remove inhibition, the neuron, the final neuron, may reach threshold and then can have an action potential. So keep in mind that circuits and relationships between different neurons are not this simple. There are usually hundreds of neurons that can converge on one neuron, or one neuron that can contact hundreds of others, but it's just to give you the idea, give you an idea about some of the common neuronal connections that we have in the nervous system. So there are other examples, including lateral inhibition, where a neuron that is activated then inhibits adjacent neurons, and feedback inhibition, where a neuron is activated and then it activates an interneuron that will inhibit it, basically with the purpose of turning off the message once it has been passed on to the main target. So it will inhibit itself. So now that you have a basic idea about the cells of the brain and how they communicate with each other, we will start to move into more complex concepts, such as the different functions that the brain can produce and is responsible for.